The correct view there.
Yeah, there it is.
Perfect. Thank you so much, Franz, and everyone really for spending the time to talk with us all about MZ.
Well,
And
thank you for
today,
being here, right? And providing
oh,
a
of course.
solution, right? Come on.
No, it's a great honor to be able to talk
Yeah.
directly to you all, and really interested in everyone's thoughts as we progress this program too. So today I'll be talking about BMN 349 in our investigational program that's studying both alpha 1 ZZ as well as MZ. So as Raynaud and Franz both presented, we know that there is different levels that drugs can work on. BMN 349 is the investigational drug that we are exploring that acts at the level of the protein. So the mechanism of action is intended to bind the mutated AAT or the ZAAT in the liver cells to enhance the secretion out into circulation, really with the goal of blocking the ability to make those disease-forming polymers in the liver. So I have one more slide that has an image here, which I think does a little bit of a better job, at least for me, I'm a visual learner. So on the left, you can see an alpha 1. And as Franz mentioned earlier, both in ZZ and in MC, you have the production of the mutated ZAAT to different levels. So without any therapy, without anything going on, we know that that ZAAT is not able to be secreted when it's supposed to be. So only about 15% of the mutant AAT is secreted. The rest is either degraded or forms these disease-causing polymers. So on the right, you can see the intended mechanism of BMN 349, and it's demonstrated there by that half oval. So you can see it binds to the mutant ZAAT, allowing it to have an increase in the amount of secretion out into the blood, which reduces the amount that's stuck in the liver and results in fewer disease-causing polymers. So I just want to focus on three main things that make this drug a little bit unique. One is that it is an oral medication, unlike some of the others in development right now. The second is this mechanism. So as we mentioned, some are working at the DNA level, the RNA level, and then we are working directly on the prevention of polymer formation. And then the last thing is that we have preferential binding to the mutant AAT over the MAAT. And that means that we can actually look in MZ. So other therapies can potentially inhibit all of the AAT, regardless if it's M or Z. But this therapy, so far in vitro, has been able to show a preferential binding specifically to the ZAAT, which is why we are really excited to be able to further explore this in the MZ population. Okay. So, apologies. You can see here, this is just a graph showing overall from ZZ to MZ. We know that ZZ is a lot less common in terms of prevalence, but then some of the risks for liver disease and otherwise can be a lot higher. I know previously it was thought to have MZ as a minimal risk in the past, but more recent research is really showing that that is no longer true, particularly as there are additional insults. So you can see that the prevalence overall of MZ is more common. And even though the risk of cirrhosis or liver scarring is a little bit lower, it's increased overall, and it's increased when you have a secondary insult. So we can really see that MZ acts as a disease modifier in individuals with coexisting liver disease caused by other causes, things like alcohol, viral hepatitis, or fat in the liver. So some general risk factors for developing liver disease are on the bottom there, and those you can find particularly MZ in terms of diabetes, obesity, or metabolic syndrome, leading to increased risk of concomitant liver diseases. So I know Franz also presented a little bit on what are some of these risks in liver disease. I wanted to just kind of highlight a couple of studies. I think he already mentioned the UK Biobank. And so on the left, we're just looking at the MZ population with no specific other coexisting liver disease. And what we see on the left is that those with MZ had almost 10% having significant fibrosis. And why we care about fibrosis in the liver is that it is directly correlated to mortality. And so we really want to be able to prevent or reverse potentially fibrosis to be able to reverse the advancement to more advanced disease. The UK Biobank study also showed that those with MZ versus MM have around a 1.7 increased risk of fibrosis and cirrhosis. So as I mentioned, that was all just looking at the MZ population, but what studies have also shown is that with a coexisting secondary insult, the risk is even greater. So in this case, we are specifically focusing in on metabolic dysfunction associated steatohepatitis, or MASH, also called fatty liver disease. So you can see the progression of MASH on this slide, starting from a healthy liver, then having some fat infiltrating the liver, and then having disease in terms of fibrosis and scarring, and ultimately potential end-stage liver disease. So what we see in patients that have MASH and MZ is that they have a 6-fold increased risk of leading to cirrhosis. Then in those with cirrhosis, MZ is associated with more advanced disease, and that means different signs and symptoms and liver transplant, as well as liver-related death. So this is part of the main reason we have focused our initial studies in MZ to concomitant MZ plus the presence of MASH. So I want to give an overview of where we are with this program. On the left, you can see we first studied this drug in healthy volunteers. So we've looked now at 6 different doses at a single dose, and then 3 different doses in multiple doses. So this data should help us move forward in terms of what is a safe dose that we think will act as it's supposed to. But unfortunately, as we know, this drug is working on the mutant AAT, and those without disease won't have mutant AAT. So it's hard to be able to predict response in people with disease when the drug is acting on ZAAT. That is why we have this study on the right. It's a single dose study, a very small study looking at 6 patients with ZZ and 6 patients with MZ and MASH. So we are currently enrolling this study in the US and the UK. And really the objective is to understand the pharmacokinetics, how the drug is processed, and what might be a dose that we can move forward in that we think is going to have the intended effect. So a little bit about this study, as I mentioned, the key eligibility criteria are on the left in terms of age. And for ZZ, we do not allow at this point the use of augmentation therapy as a phase 1 study. For MZ, as I mentioned, the main criteria are the age, the having coexisting MASH liver disease, and some evidence of liver fibrosis. You can see on the bottom the list of the different sites that are currently running this study. And really the overview of the study, it's an oral tablet taken once on that first day, and then there's safety assessments and PK blood assessments up until about a month later. There is no biopsy, and there are mobile nursing services so that the blood tests and other tests can be done at your house or at work or wherever may be most convenient, because we do recognize that participating in a trial, particularly a phase 1 trial, can be quite burdensome and take up time. So our next hope is with this data is to move into a phase 2 study in the next year. So you can see there will be two different phase 2 studies, one looking at just the ZZ indication and then one looking at the MZ indication with MASH. And this study design is called a multiple ascending dose, or a MAD study. And what that means is you start at the lowest dose, you dose for about a month, and then we look at the safety and the PK and see, is it safe to go up to the higher, to a next higher dose? So really the purpose of these studies is to find what is the highest dose we can go to in terms of efficacy that is still safe. So we hope to start these studies within the next year and share more information with you on these. So if you are interested in potentially participating or learning more about this study, there's a QR code here and a link to our BioMarin clinical trials website. You can also, of course, find us on clinicaltrials.gov. I will also say, if you are interested in sharing your thoughts on the Alpha-1 experience and MZ and liver disease, but you may not want to yet participate in a trial, Franz also sent out in one of his newsletters a research opportunity that we are working with an external group to gather thoughts and do a couple of interviews. It's an independent group from us, just to understand from the patient perspective where the disease burden is and what your thoughts are. So I would refer to the newsletter if you are interested in that. So for more information, if you have any other questions, please feel free to contact us. Our contact information is there and can be found as well on our clinical trial page. Thank you so very much for allowing me to speak with you all, and we are very much looking forward to advancing this program.
Okay, Jenny, thank